Thank you very much for inviting me. Since this is an investment-oriented section of the session, it may seem at first as though the talk that I'm giving doesn't really apply, but as we get toward the middle and end, you'll see that it really is deeply applicable. So what I'd like to ask of you now is to cast your mind back into the halls of history, back to the early days of the 20th century. In those days, there was no AMA, there was no FDA, no NIH. There was no food or drug labeling. Meat packers and food manufacturers were able to put whatever they thought was good for business into their cans and bottles and sold them to the public. Some were ethical and some not so. The public health suffered as a result. So were these manufacturers evil? Maybe. But here's the truth. Nobody had any scientific basis for knowing what was bad or for you or good for you. So, in order to fix that terrible situation, a brave group of folks at the United States Department of Agriculture decided to test these foods systematically on themselves. These people became known as the Poison Squad, and happily, since they were human, the results of those experiments were immediately and directly applicable to society and resulted in huge benefits to the public health, our health. We are still benefiting directly from those experiments. Experiments on humans? How in the world could that happen? It's illegal. Well, up until World War II, it wasn't illegal. It was typical. Because of using humans in those early days, it was possible to, in just a decade and a half, to remove some terrible compounds from food that were thought to be useful. Well, then in World War II, the Nazis decided this is a good opportunity to do human experimentation on unwilling subjects. These atrocities in the 40s led to the Nuremberg trials after they lost the war. And part of that was the Nuremberg Code resulted, which correctly favored animal-based experimentation. So the Nazi doctors went to prison, and here's what happened with the Nuremberg Code. In part, it says, the experiment should be so designed and based on the result of animal experimentation and a knowledge of the natural history of the disease or other problem under study, that the anticipated results will justify the performance of the experiment. And so we turn mostly to mice and other model organisms. It turns out that we've been using mice and other animals to further our understanding of the physical form and improve our health for thousands of years. As far back as 2000 BC, Babylonians and Assyrians documented surgical and pharmacological experimentation on animals. In our era, mouse-oriented animal model research grew from the start of the 20th century and has been almost solely relied upon for scientific research over the last 25 years. Each year, more than 100 million mice and rats give their lives for our benefit. What kind of benefits? Well, the dog gave us insulin and kidney dialysis. The mouse gave us penicillin. The monkey gave us a polio vaccine. The pig gave us heart bypass and transplant techniques. And the rat gave us a vaccine for tuberculosis. Rabbits for meningitis, and cows continue to give us the necessary components for heparin. Animals have given us a wealth of medicine. The list would be too long to go into here, of course. So why does this work? Well, because animals share basic functions and mechanisms that have been useful in identifying the physiology and disease patterns in our body, especially in more complex system of systems. The animal model has gifted us life repeatedly. And so we started the 20th century at 43 plus years of typical lifespan, and now we're well over 70. Sounds good, doesn't it? Well, unfortunately, mice and men are not the same. We may have 90% or so corresponding genomic regions, but we can all agree that there are obvious differences between us. I'm a lot bigger and in some ways fatter than mice. And so the naked eye is a good measuring tool to say, no, really in a lot of ways, we're completely different. So what are results? We see research that finds promising results in an animal model that has no therapeutic effect or even a dangerous response in humans. For instance, natural mice don't develop Alzheimer's disease. So we have to develop animal models that mimic Alzheimer's disease, but honestly, we don't really know if it's a valid model. What we can say is that the use of animal models for Alzheimer's disease has led to complete and utter failure in finding a cure for Alzheimer's. We remember the thalidomide experiments. It was proven safe in mice, rats, and rabbits in 1962, and yet created many malformations in humans, in unborn humans, and so it had to be taken out of the market. Now, there's another unexplored side to this, and that is the potential for missed opportunities, because what might hurt animals might benefit humans. An example is linezolid, which is currently being used effectively to treat tuberculosis, and is helping well over 1.5 million people. But this drug doesn't work at all in mice. It was accidentally discovered and we were able to bypass the whole animal model approach. If that hadn't happened as a historic anomaly, it would have been lost to the world. We're well aware that 1/3 of animal research doesn't translate in human trials. Now let's get to investment. The cost of each drug reaching the market has a median of $650 million. And this doesn't account for failed attempts. If we add failed attempts in, it's closer to $1.5 billion. It takes over a decade, well over a decade, to get to that one approved drug, typically. Wasted trials, time, and talent. We are in an era that's similar to the time before the printing press, but we don't necessarily realize it. In the time before the printing press, a scribe may, in his career, produce one Bible. 20 years, a Bible. And now we have scientists who may spend their career on the research for a single drug to take it through to the market if it's successful. So mice aren't humans. Lab mice are inbred and genetically identical, but we are not. We're extremely varied. Mice are exposed to a single environment, an unnatural environment in their cages, and humans have a vast envirome, which we are still exploring and understanding. So they may be raised in germ-free barriers, but as COVID-19 shows, we're not in a germ-free environment. So while humans and animals are similar, it's at best an imperfect model. So what do we do? Well, as we mentioned earlier, animal models were not the norm in the early 20th century. So this poison squad deliberately decided to poison themselves for the sake of humanity. Let's come to a more modern era. Jonas Salk, Albert Sabin gave unproven polio vaccines to themselves and their families. And Salk's vaccine was tested with a mass inoculation of school children, and it was successful. Recently, David Pritchard has been self-experimenting with the use of hookworms as a method to improve allergic reactions and decrease systemic inflammation. How can he get away with it? Because it's him, and he's decided it's important enough to do it. What is the most important about experiments in humans is that the data is predictive and usable. And 89% of self-experimentation research, the researchers obtain valuable data immediately. Positive results actually can support a hypothesis, and even negative results are meaningful and dispositive at minimum. The animal model results in results where we really have no idea if they are false negatives or false positives. This adds at least half a decade to research and tens, if not hundreds of millions of dollars of investment money, where most of the money goes into a barrel where gasoline is poured on the money and it goes up in flames. Why do we do that? The truth is because we didn't have an alternative. So what do we do about all of this? Well, we still need a whole organism to determine systemic interactions, but we can't use humans. Well, now we have 3D printed tissue. And allow me to digress for a moment. In the late first decade of the 21st century, the Methuselah Foundation was privileged to make a founding investment in a company called Organovo. Now, Organovo had the first commercially available and usable 3D human tissue printer. When I was first talking with the founder of Organovo, he and I had a wonderful discussion and talked about the potential for using fully three-dimensional human tissues to do two things. One would be to break this logjam of animal model testing by using human tissues that were architecturally and had full fidelity with organs. In this case, organ tissues such as the liver and the kidney. And then further, to be able to model disease states, to model cancer, tumors, non-alcoholic fatty liver disease, kidney diseases, and so forth. So that was the vision. By 2015, 3D human liver tissues were actually for sale, and then shortly thereafter, in a collaboration with an Australian lab, Organovo produced 3D architecturally accurate and complete kidney tissue. So for the first time, an alternative was available that was not quite human testing, but was vastly superior to model organism testing. In those early days of testing on these models, we discovered that by testing, let's call it back testing, these, let's say the failed drugs that had already gone to market. Now imagine that you'd spend $1 billion or $1.5 billion to get a drug to market. And after 6 months, people start having heart attacks on this drug. Maybe it's just a few, maybe it's hundreds, or maybe it's as bad as thousands. Well, before you know it, that drug will be pulled off of the market. We took several examples of those failed drugs and tested them on the 3D tissue models to see what they would have said if the models had been available at the beginning. And even in those early models, 85% of the time, those 3D tissues predicted failure. and failure that was genetically the same as what happened in the humans. Now, I'd like you to think about that for a moment. What if, as an investor, you could invest in a research program or a drug, a small molecule, that had been tested on human tissues that had architectural fidelity, and instead of being in vitro two-dimensional, where there is no cell-to-cell, network-to-network communication, biologically speaking, you go to the 3D, which is where we live, what we are, and then all of a sudden, it actually starts to work. That is where we are moving today. So instead of a 10 to 12 to 15-year process of getting a drug to market, and along the way, never even knowing if it's going to actually work, and by work, I mean medically, but also financially, you could cut that down, conceivably down to 5 years. And instead of $1 billion, $1.5 billion, you could cut it down to a reasonably 100 million at the outset, and maybe even less than that as time proceeds. Now, there's a side point here. I don't like killing animals. I don't know about you, but I presume that you feel the same way. In Europe, the use of experimental animals for cosmetics has been banned starting a couple of years ago, I think. So that bodes well for the 3D tissue model approach because they have no alternative. And last year, the medical apparatus in the United States, I think, said that by 2035, it will be banned here. Well, that sounds like a long way away, but if it takes 15 years to get a drug to market, you better start looking at alternatives. So as an investor, we now have a green field. The green field, it's an open field. Organovo almost failed as a business, but I am happy to report that the company is going to be doing well. In mid-September, there will be a stockholders vote, voting for a new slate of directors, which includes the founder, and I'm happy to say I'm on the slate as well. And we are going to go back to the future and refocus on these very high value potentials. So that's Organovo, which is publicly traded. With my partner, Sergio Ruiz, the Methuselah Fund, which is majority owned by the Methuselah Foundation and is mission driven by its many supporters and investors, has invested in 2 additional companies in this field. One is Volumetric, which makes what I call holographic 3D printing and have advanced microvascularity capabilities, which further improves 3D tissue modeling and makes it more has higher fidelity to human tissue. And also Vizient Bio. Vizient Bio was a spinout of Organovo when Keith Murphy, the former CEO of Organovo, left the company. Within 2 years, his company was worth about $50 million or so. It's still private. And they're working on non-alcoholic fatty liver disease using the 3D tissue models that he pioneered at Organovo, and he has full rights to those at Vizient. And they've made several breakthroughs and remember the 85% number that I gave you before, about 85% of tests on failed drugs would have been predicted. The number is confidential, but it is now much better than 85% because the technology is improving as we go along. An unexplored territory is personalized medicine. Since these are fully 3D human tissues, we would be able to take a person's genome incorporated into these 3D tissues, and we would be able to test the likely reactions of drugs on an individual patient. This will take time because of the expense, of course, but imagine being able to test oncological interventions on your genome in 3D. It would know exactly which approach to take for you. So to summarize, we need this. We simply cannot expect to improve our longevity within our lifetimes when it takes 20 years for a drug to be found out, tested in animals. Many of the companies where the testing would have worked run out of money and they die, and the intellectual property lays there and molds in the sun, rusts. So many things happen that prevent even successful things from getting to market. We need to tighten, shorten the valley of death from 15 to 20 years to 5 years. And then by the end of the decade, have human biology be explorable at the rate of coding. So by moving away from dependence on the animal model, we'll have this superior 3D human tissue model, and with right to try legislation and hopefully a streamlined FDA, the only barrier is adoption to this new paradigm. And we believe that conditions, even now with the COVID-19 warp speed trials methodology, that we will have a successful transition. The question is, how quickly will it occur? And for our part, we're putting a very significant portion of our effort and our thinking into it, so that longevity-oriented and rejuvenation, health span-oriented drugs, biologics, and other forms of medicine can more quickly get to market in our lifetimes. Thank you.